Hi and welcome to my OCR A-Level Biology revision session with me, Christine. So today's lesson I'm going to look at the mammalian ventilation. So to start us off then, we need to think about how air actually gets into our lungs. So we start with our mouth and our nasal cavity. Now, our mouth and nasal cavity are a moist surface with a hairy lining. Now, that's important because what we do is the cells in our nasal cavity will actually secrete mucus to trap any dust and pathogens that we may have breathed in. As they are in the air, as they are moving around and we take a breath in, that is going to result in potentially these pathogens or dust particles getting into our lungs. Now, we don't want that to happen, so we have special mechanisms to prevent that. Now, we have a large surface area and a really good blood supply to the nasal cavity, and that is there so it can warm the air up. As air gets warmer, it's going to expand and therefore it's going to aid the diffusion of the gases into our bloodstream. Also, it's going to increase humidity within the nasal cavity, and that's important for helping that gas exchange. Now, as we move down the trachea, what we need to understand is that those pathogens, that dust that we have breathed in, could potentially get past the hairy lining of our nasal passage and get down into the trachea. Now, if it does, we need a way of stopping the pathogens from getting into our lungs. So therefore, what we have is what's known as ciliated epithelium. Now, ciliated epithelium is a tissue, and a tissue is a group of cells working together with a similar function, which would be to trap the pathogens and prevent them from getting to our lungs. So therefore, it has to be made up of many cells. In this case, we're going to start with our ciliated epithelial cells. Now, ciliated epithelial cells have cilia. These are these projections at the top of the cell, and they're important because these projections beat in a rhythm. Now, when they beat in a rhythm, what that does is that moves the mucus, which has trapped this sticky substance, which has trapped the pathogen, it moves it in a way to take it away from the lungs. Now, it beats. That movement occurs because of the cytoskeleton. So it's important to know we are talking about eukaryotic cells. Eukaryotic cells have the cytoskeleton, which is more complex. As you can see in my diagram, it has a 9+2 formation. So there are 9 parts to it that go around in a ring and 2 central parts. Now, that's just important for you to note as a distinguishing feature if you were comparing this to a prokaryotic cytoskeleton, which is much more simplified because those cells are smaller. It's not necessary to have this complex setup, whereas in eukaryotic organisms it is. So the 9+2 formation of that cytoskeleton shows that we are in a eukaryotic cell and it is more organized. Now, I did say that the ciliated epithelium was a tissue, and that tissue is made up of different cells that are working together to have the same function. Well, the ciliated epithelial cells beat, but the goblet cells are the ones that secrete the mucus, the sticky substance that's going to trap the pathogen. So the mucus is actually made up of water, carbohydrates, proteins, and triglycerides. So this is a substance that is secreted through the process of exocytosis, and that will then be able to trap those pathogens, and then we will be able to have our ciliated epithelial cells beat that mucus and any trapped pathogens or dust particles away from the lungs. So it's important that we note when we breathe in, the air is going to make its way through the opening of the mouth and the nasal cavity. Now, obviously, we can close the mouth, so the nasal cavity is the one that remains open all the time. So air should be constantly coming in and be removed from the body, and that is necessary. So we need to have an opening, an airway that remains open the whole time. So therefore our trachea is our first part of our airway moving down into our thorax region. So to make sure that the trachea remains open, it's actually made up of cartilage rings. Now those cartilage rings are made of two fibrous proteins, so collagen and elastin fibers. Now remember, collagen is a strong substance. It's a fibrous protein. It's strong, but it's flexible. And elastin fiber has the ability to stretch and recoil. So it's important that we understand that this really strong fibrous protein ensures, with these cartilage rings, that we are not actually able to shut the airway. So if you haven't already done so, do check out my video where I dissect a pig lung, and that will show you just how strong these cartilage rings are and where they are located. So what we're going to look at then is how the air comes in through the nasal cavity, makes its way down the trachea, and then it branches into what's known as the bronchi. Now bronchi is plural, bronchus is singular. So if it moves down into one of the lungs, it would be moving down the bronchus. We're talking about the plural, the two of them, so therefore it's the bronchi. The bronchi will then actually lead on and branch more into what's known as the bronchioles. So the trachea branches to the bronchi then branch to the bronchioles. And in the bronchioles, it's really important, again, linking back to our other modules, to understand what's known as the expulsive reflex. Now, if we have breathed in air and unfortunately there is some form of dust or there's some form of maybe a bit of fluid has got into your trachea, we don't want any of that getting down into our lungs. So we have what's known as the expulsive reflex. This is a non-specific immune response in which what's going to happen is we need to remove this unwanted or harmful substance in a very coordinated way. And that's through, for example, when you breathe in dust, your smooth muscles in your bronchioles will actually constrict and therefore restrict the airways. You could cough. If there's a bit of liquid, then you can cough, and that will help to remove that out of the lungs. So it's often a time when you are taking something in and maybe you've breathed it in and it's, instead of going down, it's a food substance or a liquid, instead of going down into your esophagus, a bit of it has ended up in your trachea. Then by you coughing, that should help to bring the substance, that harmful, unwanted substance, up and out. That is part of your non-specific immune response, and it's an expulsive reflex that you do. Because the key thing is, we do not want any unwanted or harmful substances, even fluid, getting down into our alveoli. Our alveoli are the site of gases exchange, and the alveoli are the place where we want to maximize the exchange surface. So therefore, we need to ensure there is nothing that is going to hinder that gaseous exchange that happens at this exchange surface. So how do we actually ventilate then? So then we have to look at, well, our lungs are encased in our rib cage. So we have these ribs which completely encase and protect our lungs. And in between our ribs, there are intercostal muscles. Now, there are external intercostal muscles on the outside and internal intercostal muscles on the inside. And muscles work antagonistically. We know that muscles can contract or relax, and if we have ones that contract, the opposite, the antagonistic muscle, needs to relax, and that therefore allows for this movement of our rib cage and also the movement of our diaphragm. So our diaphragm is a muscle which is connective and is in a dome shape. So if we look then at this gaseous exchange and we want to talk about what happens when we get down to the alveoli, we need to note then that we have what's known as squamous epithelial cells. So before we had our ciliated epithelial cells, and now at the alveoli, we have these squamous epithelial cells. They are squashed cells, which are going to have a short diffusion distance, therefore maximizing the exchange of the gases, oxygen into the transport system and carbon dioxide out. Now, they also have what's known as a surfactant. So surfactants are layers of phospholipids which are on the surface of water, and they stop the alveoli from collapsing. We want to maximize the surface area that is exposed so we can maximize the diffusion that can occur. So we need our alveoli to remain open, these air sacs. And our ventilation then, the bringing fresh air in and removing stale air that we do, along with our blood supply, where our capillaries run all the way in between where our bronchioles and alveoli are, is necessary to maintain a concentration gradient so that our oxygen will diffuse from the air that's breathed in into the bloodstream, and the carbon dioxide will diffuse out of the bloodstream and into the alveoli. So how do we ventilate then? What is the process that allows for ventilation to happen? Well, the first thing we need to do is we need to bring our rib cage up and out. So you can quite clearly see here that when I am sitting, and if I was not doing anything, what I would need to do to allow for the air to be brought into my lungs is I need to lift my rib cage up and out. Your lungs are attached to your rib cage by a connective membrane. It's called the pleural cavity. And what happens is when the rib cage moves up and out, because of the muscles contracting, that's going to take the lungs with it. So what you have is when we are inhaling, our external intercostal muscles are going to contract. Our diaphragm is also going to contract. So what we have done is we've lifted our rib cage up and out, and our diaphragm has moved down. That is going to increase the volume inside the pleural cavity. Now, because it's increasing the volume, that's going to decrease the pressure and make sure that pressure is lower than the atmospheric pressure. That will cause air to flow into our lungs. Now, if we want to breathe out, if we want exhalation to occur, we need the opposite to happen. Our external intercostal muscles are going to relax. Our diaphragm is going to relax. This time, elastin fibers, those fibrous proteins which were in the alveoli, in the bronchioles, in the bronchi, and in the trachea, they are going to recoil back to their original position. So we have these elastin fibers that we can stretch, we can bring and pull out, we can stretch them. And if we are no longer pulling them in the stretched position, if we release that when the muscles are relaxing, they will naturally recoil back to their original position. And when they do that, that is therefore going to decrease that volume, therefore increase the pressure and push the air out. So when we are ventilating our lungs, what we are doing is we are ensuring that the air comes in, which is full of oxygen, and the carbon dioxide is actually removed. So we need to ventilate our lungs so that we can bring in fresh air, which has more oxygen, so that we can continue to do aerobic respiration, and we can get rid of, excrete, the waste product carbon dioxide. So how do we visualize this? How do we see this? Well, we can actually measure a person's lung capacity using what's known as a spirometer. Now, when you use a spirometer, you would get the person to be connected to a spirometer, which can be in many different forms. It depends if you have access to one at school. It may be connected to a water chamber. It may be a digital one. The key thing is that you know that it's called a spirometer, and it's going to measure the person's lung capacity. Now, we normally display it with a data logger. So what you can see here is a spirograph, and what we're going to look at is what has happened to the lung volume. So there's some key names that you need to know. Now, the first one, when you are breathing normally, is known as your tidal volume. So here you can see my tidal volume where I breathe normally, and that is from peak to peak and trough to trough. If I take a deep breath in, that is called my inspiratory reserve volume. So this is the maximum air that I can take in if I take a deep breath in. So I am bringing more air in than my normal breathing, which is my tidal volume. If I then take a deep breath out, and I really try and force that air out of my lungs, that is called the expiratory reserve volume. So the air, the extra air that I can push out of my lungs, that expiratory reserve volume, that actually comes from forced expiration. Now remember I said there are external and internal costal muscles. Well, normal breathing in and out at rest is with your tidal volume, where your external intercostal muscles are contracting and then relaxing. If I want to force my breath, if I want to force the air out, like really push it out, then what I'm doing is my internal intercostal muscles are contracting and my abdominal muscles are contracting. What they both do is they push the diaphragm higher and they'll bring the rib cage in further, again affecting the volume. If we affect the volume, we affect the pressure, we can force more air out. So if we looked at the inspiratory reserve volume along with the expiratory reserve volume, that with the tidal volume is actually called your vital capacity. So the vital capacity goes all the way from the peak of the inspiratory reserve volume right the way down to the trough of the expiratory reserve volume. So make sure you know these definitions. Now, you never completely clear out the air inside your lungs. Remember, because we have the surfactant, because our alveoli never collapse, we therefore have what's known as a residual volume of air that stays inside the lungs. So that is known as the residual volume. And if we added the residual volume and the vital capacity together, that would give you your total lung capacity. So what they may do in the exam is they may actually give you a spirometer and data logger set of results, and then they ask you to use that to interpret what is happening. So we're told here that the student initially was at rest, and then that student changed to carrying out some strenuous activity. So they've given us some information here. So we can see first of all that if we look, here is my tidal volume. Here's my normal breathing. This is my normal breathing, peaks and troughs in that tidal volume. And then the strenuous activity has started to happen because I'm needing to bring more of the air into my lungs because I need to ventilate more. So I can see straight away that in this question, the person was probably just at rest until about 35 seconds. So it's important when you're given information in a graph like this that you know how to interpret what it is that you're seeing. So that's the first thing I would do, is I would identify, right, where's my tidal volume? I know that's the person's normal breathing, and that will help me to identify where they were at rest. Now the question actually asks, calculate their ventilation rate. Now when we do ventilation rate, we want to understand that tidal volume. So what you would then need to do is say, okay, so it's asking me for my ventilation rate. Now I know my ventilation rate should be the number of breaths that I take per minute. So if I'm calculating my ventilation rate, I'm looking at how many breaths have I taken per minute. So in this case, I can't tell for the full minute because I've started doing some exercise, and therefore, because I've started doing some strenuous exercise, it would have an effect on my ventilation rate. So what I would then do is I would actually take a reading up to the 30-second. What I would do is I would take a calculation and see that there are 6 troughs in 30 seconds, and I know that there are 2 30 seconds in a minute, so I would therefore do 6 times 2 to tell me that my ventilation rate would be 12. So it's important that you're able to identify what it is that they are asking you in the question, and that you can then take a reading based on the information that they've given you, because it's about you applying your knowledge. So I hope you've liked this video, and if you have, then please do click the like button and subscribe to my channel. Also, if you haven't done so already, check out my revision platform, www.aiqchat.com, to help you with your revision.